Okay, so what are we talking about today? Today we're going to focus on a couple of things as we look at animal development. We're going to look at how does fertilization occur? What are the stages that we're going to see in embryonic development? And we're going to talk about those in quite some detail. So we're going to focus on cleavage, gastrulation, and organogenesis, right? And then we're also going to look at what are some factors that have been shown to influence embryonic development, right? How does this process occur? Okay, and what are the things that are potentially really affecting it? So starting from the beginning, right, so here kind of we can graphically look at this, right, so we're really talking about sperm and egg, right, that is really looking at fertilization. We form this zygote, right, the fertilized egg, and then we're going to go into looking at cleavage, right, and so in cleavage we can see, and we'll look in a lot more detail, we're getting rapid cell division, okay, forming this blastula, right, then we're going to go through gastrulation, okay, then we're going to go through organogenesis, where essentially the basic outline of the organs have formed kind of at that point. So starting from the very beginning, let's look at fertilization in some more detail. So what's happening in fertilization, right? So the first thing here is you can see that the sperm, right, is coming to the egg, right? And if you notice, there is something called an acrosome on the tip of the sperm, okay? The acrosome really, this is a special vesicle that sits on the tip of the sperm, okay, that's going to contain enzymes, right? And so it has these enzymes, okay, and those enzymes are going to go ahead, right, and you can see them being released here, and they're going to go ahead and digest, right, this kind of jelly coating that exists around the outside of the sperm, okay? And what you can see too then at this point, right, is you get this acrosomal kind of process, really, if you will, that extends, right? So call this the acrosomal process. And this is going to extend, right? It's going to interact with these receptors, right, on the surface of the egg cell, right? And this is acting kind of like this lock and key fit. Okay, and then what we can see as that happens, right, then the nucleus, right, from the sperm will go ahead and start entering the cytoplasm of the egg cell. Okay, so as this is happening, a couple of things are going to happen. One thing that's going to happen is sodium ions are released, right? So sodium ions cause the membrane to be depolarized, and this is acting as this fast block to polyspermy. Right, because the egg cell doesn't want to go ahead and be fertilized by multiple sperm. There's also, right, a slower, kind of more permanent block to polyspermy, and you can see this as well, right, where here you have these cortical granules, okay, and so what these are going to do, right, is these are going to go ahead and release enzymes and some other materials that are going to cause this layer, right, this vitelline layer, to go ahead and lift up and harden. Right, and so as this kind of lifts up and hardens, it's going to create essentially this longer block to polyspermy. Okay, so you're going to get this longer block to polyspermy because this initial membrane depolarization only lasts kind of like maybe for a minute, and this allows you to kind of get going this longer block to polyspermy. That's going to happen, right, as you're kind of changing like the egg surface in this way. Okay, so this is kind of the first step, is thinking just about membrane, is thinking about the sperm fusing with the egg cell, right, and this is going ahead and causing fertilization. Okay, so kind of this first step as we're really looking at embryonic development. So after fertilization, you have a zygote, right, and so this is after the sperm and egg have fused, right? You have a fertilized egg, which becomes a zygote. And so if you notice, okay, so what we're going to see here in the next phase of embryonic development, right, in early animal development, we have this cleavage phase, and the cleavage phase is really marked by rapid cell division. Okay, and what we're going to see here, right, is that very quickly you're going from 1 cell to 2 cells to 4 cells to 8 cells, right, and then finally we're going to end up with a blastula. And something to notice here that is the amount of cytoplasm is not increasing. Okay, and so what we're seeing is that we're really going through S phase and M phase, right? And we're really skipping G1 and G2. It's not growing. We're just taking and dividing up the cytoplasm. Okay. Also, if you notice too, this is an example of kind of what's happening in frogs, but this is happening in frogs and other animals as well. We have what's called an animal pole and what we call a vegetal pole. And if you notice, right, at the vegetal pole, this is where kind of the yolk is concentrated. And the yolk, right, is containing kind of the nutrients and other things that are going to be sustaining this zygote, right, as it, well, it's not a zygote anymore, but it's sustaining kind of the growth as it's in development. Okay, so it's containing a lot of nutrients. And then what you can also see as well, too, right, as we go to this blastula stage. So this is really after kind of like 5 to 7 divisions that you're reaching this blastula stage. And when you reach this blastula stage, you notice what you have, this fluid-filled cavity, and this is what is called a blastocele. So this is really just fluid in here, okay, and it's surrounded by all of these cells. And this really kind of marks the end kind of cleavage. And some things that are going to be important to kind of notice is that where the yolk is distributed is also going to kind of affect how the development is going to happen, right? So that's just kind of something to think about here in terms of what's happening with cleavage. In mammals at this stage, the blastula is known as a blastocyst. And what we're seeing is a couple of things, right? So what's really key to actually notice is that within this, there's what are called these cells that are called the inner cell mass. And this will become the embryo. What's very interesting, right, is that these cells are what we call pluripotent. Okay, which means that they can develop into all different tissue types, right? And these cells are what are the source of embryonic cell lines. Oops. Right, so these are often what people are talking about when they're talking about embryonic stem cells, right? So these are going to be the embryonic stem cell lines. Okay, because they have the ability to go ahead and develop into many different tissue types. Okay, and so this is really important in terms of thinking about not only development, but also how these cells can potentially be used or are being used in terms of research and other things as well. Okay, and this is different from what are called totipotent stem cells, because these cells, right, can go ahead and develop into an entire organism. Whereas what we're looking for with these pluripotent stem cells, right, is that their cell fate hasn't really been totally specified at this point, right? Because we're only really after 5 to 7 divisions that we're at the blastula stage, right? So that at this point they're not all differentiated, and they're going to go ahead and differentiate later into specific cell types. And so this is going to be important to look at in terms of embryonic stem cells. And so as we're looking at this, right, you can see that out of this blastocyst, right, what's happening, right, is you can take these pluripotent inner mass cells and they can go ahead and differentiate into these different stem cell types, right? And again, these are unipotent at this point because they cannot, their cell fates have been determined, right? If these are cells that are involved, that are nerve cells, they cannot go ahead and now differentiate into cells for the immune system or for the circulatory system. Their fate's already been specified, okay? Whereas if you look back here at this totipotent stage, right, at that point, it can still develop into an entire organism, okay? And so this is something to kind of look at. So what we're going to do is we're going to take a look at an animation now that it's going to give kind of a little bit more detail in terms of how embryonic stem cells are developing, and then how they can actually be extracted and then going ahead and grown in culture to differentiate into different cell types.
Embryonic stem cells, the most pluripotent of all stem cells, are derived from embryos generated by in vitro fertilization. When fertilization is successful, the sperm head bearing the nucleus enters the egg, while the tail is left behind. The egg divides first into 2 cells, then into 4. With more divisions, a multicellular ball of cells known as a blastocyst is formed. If you could look into the blastocyst with X-ray eyes, you'd discover that it is a hollow ball made up of 2 cell types, an outer layer, the trophoblast, which will eventually form the placenta, and an inner cluster of cells known as the inner cell mass. The embryo is derived from the cells of the inner cell mass. It is possible to pick up these embryonic stem cells with a pipette and transfer them to a petri dish for culturing. Under appropriate culture conditions, these embryonic stem cells divide or self-renew, and the cell mass grows. Sometimes pockets of cells will stop dividing and begin to differentiate spontaneously. Groups of cells may develop properties of mature bone cells or of pancreatic cells. Others develop into muscle cells that can contract, and still others into nerve cells. Because they have the potential to become a wide variety of specialized cells, embryonic stem cells are described as pluripotent. Pluripotency is one of 2 key features of embryonic stem cells. The second key feature of embryonic stem cells is their ability to divide or self-renew indefinitely while retaining their undifferentiated pluripotent state. Small groups of cells can first be grown in the petri dish, but as the numbers increase, the population can be further expanded by growing in larger tissue culture flasks. You can tell that the cells are growing because the tissue culture medium turns from pink to yellow-orange, indicating a drop in pH. Cells from a single flask can seed many flasks. In this way, unlimited numbers of undifferentiated pluripotent stem cells can be produced.
The next phase in development is gastrulation. And what the result of gastrulation is going to be is going to form a gastrula that's really going to have 3 germ layers, right? And so these 3 layers are going to go ahead and form the ectoderm, the mesoderm, and the endoderm. And we'll talk about those a little bit more in terms of what they, because I mean these 3 layers of cells will develop into all the cells within the body of that organism. So we're going to take a look at 2 examples. First we'll look at the sea urchin, and then we're also going to go ahead and look at how this is looking at within a frog. So what you can see here, right, is you have the vegetal pole again, and that's where the yolk was concentrated, right? And so then you have these mesenchymal cells, right, which are breaking off, okay? And as they're breaking off, you're also seeing here this vegetal plate kind of is invaginating, right? It's starting to kind of go ahead and pinch in, if you will, okay? And then you have these Philopodia cells, which are going to go ahead and extend out. And as they do, you're starting to form, which is key here, is what's called the blastopore. Okay, and so the blastopore, right, well, and before that you form the archeneron, and as you did this, so this is going to become essentially the gut, right? So that's the primitive gut, if you will, and you're forming this blastopore, right? So now you have this opening to the outside, and as you can see, right, as that extends out, you'll then form a second opening, okay? And so this is forming your digestive tract, right? So you have the opening that goes from one end to the other at this point, okay? And something to note, right, is that sea urchins are what we call deuterostomes, and they are referred to that, right, because the mouth, right, forms from the second opening. And we are deuterostomes as well, right, as are other chordates, right, things that have a notochord. Whereas things, right, that go ahead and their mouth is formed from the first opening are what we call protostomes. Okay, so this is looking at sea urchin and kind of how these 3 germ layers are developing, right? And then ectoderm, mesoderm, and endoderm will develop into all the different cell types that we'll see within an organism. And we can also go ahead and see this as well within the frog, right? And so within the frog, A couple of things to notice as well, right? You're seeing something very similar in terms of you have these three different cell types, right? So you have the endoderm, the mesoderm, right? And then you have the ectoderm, okay? And you're seeing them go ahead and migrate, right? And you're going to go ahead, right, and see these eventually, right, you're going to see these two openings form, okay? And as these two openings form, right, that's going to again create this digestive tract, okay? And that's something to kind of think about that's important. And we're also going to talk in a little bit more detail later kind of the importance of the dorsal lip in terms of what's happening there. So all of this is going to result in really these three germ layers, because again at this point, right at the end of all of this, okay, you're going to have these three different layers of tissue. You have the ectoderm, the mesoderm, and the endoderm, right? And then you have the archeneron, which is essentially acting as kind of this primitive gut, because that will develop into the digestive tract. And so you can see, right, the ectoderm, because ecto means outside, so you're seeing this as the outer layer of the embryo, and that will develop into such things as, right, the epidermis, the nervous and sensory system, the pituitary gland, and the adrenal medulla, right, the jaws and teeth. And you're seeing meso, right, is the middle, and so that's going to develop into most of the different body systems, such as the skeletal and muscular, circulatory and lymph, excretory and reproductive, the dermis of your skin, and the adrenal cortex, right? And then the endoderm is really the inside, and so that's going to be creating the epithelial lining of the digestive tract, the linings of all the other things, respiratory, excretory, and reproductive tracts, and then the thymus, thyroid, and parathyroid glands. So we're seeing all these, right? So all these cells have now, their fates are starting to differentiate in terms of what they will go ahead and become. Okay, and so let's go ahead and take a look at an animation that's going to help explain this process a bit further.
If we look at a cross-section of an embryo of the frog Xenopus, we can see that at this point it is a ball of cells with a fluid-filled cavity. The cavity is the blastocele, and the embryo is currently in the blastula stage of development. A blastula contains large yolk-filled cells at the vegetal pole and smaller cells at the animal pole. The three colors represent the three tissue layers that become defined early in embryogenesis. Yellow indicates endoderm, red indicates mesoderm, and blue indicates ectoderm. At the beginning of gastrulation, a few surface cells called bottle cells constrict at their apical ends and expand at their basal ends. These cells move into the interior of the embryo, followed by other surface cells. We can track the movement of cells into the embryo if we add dye to a few surface cells. The movement of cells into the embryo creates a lip, called the dorsal lip, over which sheets of cells continue to move inside. At the same time, the ectoderm extends around the embryo surface in a process called epiboly. As gastrulation proceeds, a cavity, called the Organogenesis, forms while the blastocele progressively shrinks. The Organogenesis is the primitive gut and is completely surrounded by endodermal tissue. The endoderm at the roof of the cavity originated from the outside of the embryo. The cavity is continuous with the outside via the blastopore, which eventually becomes the anus of the animal. As the ectoderm extends around the embryo, another set of bottle cells forms. These cells migrate into the embryo, and other surface cells follow them, creating the ventral lip of the blastopore. By the end of gastrulation, the ectoderm has surrounded the embryo. Endoderm lines the inside, and mesoderm lies between the two. Additionally, the fates of specific regions have become determined. The endoderm gives rise to the digestive and respiratory tracts and associated structures. The mesoderm gives rise to the skeleton, circulatory system, muscles, excretory system, and most of the reproductive system. The ectoderm gives rise to the skin, sense organs, and nervous system.
So during organogenesis, what we're seeing is that parts of all three cell types are really going ahead and starting to develop into these rudimentary organs. And what we're going to look at is kind of a classic example is looking at the development of the notochord and of the neural tube. And so what you're going to go ahead and see is, right, so these cells, right, are part of the ectoderm that are going to go ahead and form the neural plate, right? Those are ectoderm cells. And what they're doing, right, is you can see them folding in. And as they're folding in, they're creating this neural tube. And this neural tube, this is going to go ahead, right, and become the central nervous system. Okay, and this will be, right, the brain, the spinal cord. Okay, and as you're seeing, right, very quickly, these cell types are developing, well, along two axes, right, because this is going to run along the anterior-posterior axes. And that's another thing, too, that's very interesting when we're thinking about development. All of the signaling that's really happening to specify cell fate, right, because if, as we remember, right, within a multicellular organism, right, all cells, right, have the same genes. And it's by controlling gene expression that we can go ahead and decide which genes are going to be on and off. And that's what's going to go ahead and really specify cell fate. And so one thing that we're going to definitely want to look at here later within this is what are some of the things that are going ahead and controlling cell fate, right? And so let's kind of go ahead and jump into that, right? So again, with organogenesis, we're developing kind of these primitive organs at this point. So one thing to look at, which is very interesting, is that well, this is an organism called C. elegans, right? And it's a nematode, right? Kind of a worm, if you will. And what's interesting about C. elegans is that they've fate mapped all of these cells. And the way that they've done that is essentially by going ahead and taking and ablating, so going ahead and using a laser to destroy cells. And then by doing that, they can see and track those cells and see, okay, what do they develop eventually into? And what they've done is they've gone ahead and they've tracked every single cell within the C. elegans in terms of what does it become. Okay, and that's a little over 900 and something cells, right? And so they can see very early, right, based upon, well, this cell started here and we tracked it all the way through and it became skin cell, it became nerve cell. What did all these different things become? Okay. What's also interesting is that a little bit over 100 cells are actually killed off, right? And that's something that's interesting as well, right, is that within development, you make often excess cells to kind of start this out. So, for example, we make many excess nerve cells, and these are killed off via a process called apoptosis, right? It's also known as programmed cell death. And this is a mechanism, right, that exists within the cells, and you're going ahead and executing a molecular pathway that's going to cause the cell to kill itself off. Okay, and so that's something interesting to in development as well, is the fact that extra cells are produced, right, and they're going to be killed off. So there are two kind of classic experiments that were kind of involved in terms of looking at how embryonic development is affected by kind of signals, because again, we're really thinking about how is gene expression regulated in some of these cases. So one thing that I talked about before that I said was going to come into play was this gray crescent. And so one thing that it's important to kind of look at, and so there's a classic experiment that Hans Spemann did, right, in 1938. And what he did was this, right? So he went ahead and said, okay, I feel like in some ways he thought that the gray crescent was really controlling a determination of cell fate, and he thought that he could manipulate the embryo to see if that was the case. And so normally what happens when the gray crescent is distributed in this way, you get normal cell cleavage, right? And it develops normally. Well, what he saw was this. So what he did was he went ahead and he took and he tied a thread, right, around the egg cell, and by doing that he went ahead and restricted where cleavage would happen, right? And so that what he found was by tying this thread, right, the gray crescent was not able to kind of spread, if you will, right, across the whole cell. Okay, and since the gray crescent was restricted, he found that the gray crescent, right, so if it contained the gray crescent, they developed normally, right, all three of those. But if it did not contain the gray crescent, it did not develop normally, right, and it was then at that point became like an abnormal embryo that was lacking some of these dorsal structures, right, the back structure. Okay, and so that was a first kind of, a kind of a hallmark experiment that's indicating at this point that there's something contained within the gray crescent, right? Some sort of cytoplasmic molecule, something that's going ahead and signaling, right? And that's involved in terms of providing signals in terms of differentiation of your cell fate. There's another thing to look at as well, which is embryonic induction, and that's basically talking about, in the sense that, right, that cells can go ahead and they're essentially sending out molecules or signals that are going to affect the fate of other cells, right? And typically that's going to involve turning on or turning off specific genes. And so what they went ahead and did is they went ahead and they said, okay, we're going to take the dorsal lip, which we know to be important in notochord development, right? And take that, and we can go ahead and we can take one from a donor embryo, right, and put it on another one, right? And they use different pigments so they can track it. And then what they saw was this, right? So they saw by going ahead and placing the second dorsal lip that they actually developed a second, right, notochord, right, and a second neural tube, okay? And that these, right, that what they're seeing is that the notochord, right, which is formed from the dorsal lip, right, contain those pigmented cells, and this neural tube, right, contain mostly non-pigmented cells. And so that starting to think that with embryonic induction, right, that these cells are going ahead and sending signals to cause the fate of other cells to be specified, right? And that's kind of another kind of hallmark experiment here, where again, thinking back to gene expression, right, this is all tying to gene expression and the fact that there's signaling going on here, right, to specify cell fate, right? Because again, we started with a single cell, right, a single, well, really two cells, sperm and egg, that became a fertilized egg, right, which became that zygote, and then those cells all started to divide. Right, and those cells divided, we ended up with 3 different germ lines. We ended up with the ectoderm, endoderm, and mesoderm, right? And then all of those cells are starting to differentiate and form all of the different cells within an organism, right? And it's really this kind of complex orchestra, if you will, right, that's being kind of conducted by gene expression, right? So that each cell, right, because I mean, when you think about the complexity of the fact that, okay, what's left and right, you know, so that the heart ends up on the correct side of the body, that the lungs end up in the right place, right? What's front and back, you know, and it's a very complex process, right, that's happening all through, you know, this regulation of gene expression. So the last thing that we're going to look at is what is called homeotic genes, also known as Hox genes. And what's interesting about these specific genes, right, is that they can contain 180 nucleotide sequence that's conserved kind of between all of them. And what they're really doing is these are really kind of these master genes. Well, in this 180 nucleotide sequence is what we call a homeobox. And what these are really doing is these are kind of master kind of control genes, if you will, that are specifying developmental patterns. Okay, and so you can see that it's in terms of, and that also very interestingly enough, right, is that there's a lot of conservation of these genes across organisms, right? So that, you know, the gene that you're seeing here, right, in the mouse embryo is very similar to what we're seeing in the fruit fly, okay, in terms of these Hox genes. And so these are really specifying this kind of pattern of development, right, in these groups of cells, okay? And so again, right, thinking all back to kind of like gene expression, right, and how we're using these to kind of turn specific genes on and off, right? And, you know, oftentimes, right, in the case of these Hox genes, you know, if a Hox gene gets turned off, right, it's going to cause a body part to arise in the wrong place and things like that. And so again, ultimately thinking about this as an orchestra, you know, where gene expression, right, Hox genes, all these different types of things are regulating this really complex process.